Hello, this is Mrs. Becker, and welcome to our first by 12 video. The idea behind these videos is that you can use them as a bit of a resource to guide your notes throughout the semester. You can revisit them as many times as you want. You can access them on my website or on my YouTube channel. As you're watching, feel free to pause, rewind, watch again. And for each video, there will be a guided note that I'll give you in class that'll give you a bit of a direction of what I want you to focus on or what are kind of the big ideas as we go along. All right, let's get started. So today's idea or today's video is about cells and cell structures. Now, obviously, with the cell and especially the inside of the cell, it's very hard for us to see that with the naked eye. They're just so small. So really the study of the cells started with the invention of the microscope. As biologists became able to use the microscopes, get even more magnification, they were able to find out more and more. In the late 1830s, there were two German biologists. Now, there was one who was studying plants, and that's Schleiden, and there's Schwann, who was studying zoology, or the study of animals. Now, both of them realized as they started to talk that what they were looking at underneath the microscope looked fairly similar, even though one was looking at plants and one was looking at animals. So they realized that all organisms, regardless of plants or animals, were composed of these cells. So their discoveries are now generalized as, and this is a phrase, honestly, you will hear so much in biology, cells are the building blocks of organisms, or cells are the building blocks of life. Now, it did take also another person, a German doctor by the name of Rudolf Virchow, and he did a lot of research as well, and he tons of research about cancer and pathology. He actually is one of the first person to have seen a leukemia cell, but he said that cells come from pre-existing cells. So now it's these two statements that really make up the cell theory. One, cell is the building block of life, and two, cells come from pre-existing cells. Now, how do we take something that's just so small and find out what each of these individual compartments or organelles do? Well, we do something called cell fractionation. And what we do is we separate out the major organelles. And you take the tissue and you put it into a fluid. And then you essentially put it in a blender and just blend it up. And it kind of spits out all the stuff that's inside. And then you use what's called a centrifuge. And I don't know if you've ever been to a fair and you go on that ride, the Gravitron. The centrifuge is kind of like that. It spins and spins and spins. And using gravitational force, it removes things of certain densities. So depending on how long the solution stays in there, you'll get a certain pellet, and it becomes this pellet down at the bottom of the test tube, and you can remove that, and then within that first pellet is just the nuclei and some of the cytoplasm and that type of thing. In the second pellet, you're going to find mitochondria and chloroplast. Then if you leave it in for a full hour, you're going to find the density of the plasma membranes, and then at the very end, after 3 hours of spinning and spinning, oh my goodness, can you imagine being on a Gravitron that long? You're going to find the ribosomes. And that's how scientists were able to really specialize in one part of the cell and find out exactly what it did. Now, there are two main types of cells. There are your prokaryotic cells, and those are your primitive or simple cells. These cells don't contain a nucleus or many organelles. They have some organelles. They'll have your ribosomes. They still need to make protein. They sometimes have cell walls. They'll have their membranes. And then they often will have cilia or flagellum. But essentially, it's a very basic or primitive cell. If you remember back to Bio 11, most of the prokaryotic cells you're going to find in kingdom, or all of the prokaryotic cells, you're going to find in kingdom Archaebacteria or kingdom Eubacteria. The other type of cell is your eukaryotic cell. So here is an amoeba. As you can see, the amoeba is a little more complex. You have some specialized compartments in here, and it has a nucleus and lots of different organelles to function as efficiently as it can. Now, these eukaryotic cells are in kingdom Protista, kingdom Fungi, kingdom Plantae, and kingdom Animalia. On your note sheet, I have asked for you to draw the animal cell. This probably would be a good spot for you to pause the video and do your diagram. And then as soon as you're done your diagram with all the labeling, you can turn it back on and follow along. Now, some of the things I do want to point out to you, though, is the outside of the cell, the cellular membrane. It is the outer wall of the cell, not a cell wall. That's just in plant cells, but your cellular membrane, that's the outside, that's the outer structure of your cell. And then you have the big organelle in the middle, which is your nucleus. Your nucleus is kind of your brain of your cell, your control center. That's where you're going to have your DNA. But within your nucleus is this nucleolus, which is a dense part of the nucleus, and that one focuses mainly on RNA, and we will go into more detail in a bit. Coming out, you have your endoplasmic reticulum. Now, we like shortcuts here in science, so we decrease that to ER. We have two types of ER. We have our rough ER, and that's the rough ER is the one that has the dots all over it. Those are the little ribosomes that are attached. And then we also have our smooth ER, and that is ER that will, again, it'll go back and forth, very similar to the rough ER, but it doesn't have ribosomes on it. So smooth ER is mainly for lipid production, not protein production like the rough ER that has the ribosomes on it. You also have your Golgi apparatus, it's called here, but it could also be called Golgi body or Golgi complex. Those are two other names. All three things are exactly the same. But your Golgi complex will receive the proteins or lipids made and then will package them up, because at that point they need to probably leave the cell to go someplace else, someplace that they were meant to go. In addition, you have your mitochondria. Your mitochondria are your powerhouse of the cells. They're the ones that create that energy for cellular functioning, so the cell can do all the amazing things that it does. You also, on your cell, you'll notice this tail looking thing. This is a flagellum, and it's for motion of a cell, so that the cell can move around, but you can also have some cells that move around with cilia. You also have your cytoskeleton inside your cell. In your animal cells, you have three parts or three types of a cytoskeleton structure. You have your microfilaments, you have your intermediate filaments, and your microtubules. We'll go into more detail later. You also have these lysosomes. Here's another lysosome over there. The lysosomes kind of are the recycler or the garbage taker outer. They will go and they will, through the process of phagocytosis, they will engulf something that needs to be digested, and they have digestive enzymes inside them. They're kind of like a Pac-Man, and they'll capture that ghost and then they'll digest it or recycle it. So now we're going to go into quite a few of the details of each of the organelles. Oh, no, sorry, we're going to start with the plant cell first. Sorry, the next thing, we are going to take a look at plant cells as well. Plant cells are very similar to an animal cell, but there are some very important differences to note. So these are not in a plant cell. You won't see a plant cell with lysosomes. You won't see a plant cell with centrioles, and they will lack a flagellum. Now, there are some plant cells, specifically sperm, that will have a flagellum, but most of them won't. One of the things that they do have that animal cells don't, and that's this lining right here, that's that cell wall. And the cell wall is typically chitin based, and it has structure to it, and it is kind of like the skeletal system, you would say, of the plant cell. Another big difference, no pun intended, is that your plant cell has a big vacuole. And that's because the plant cell needs to have water. This prevents the plant cell from kind of collapsing in on itself. When you see those wilted leaves of plants that haven't been watered for a while, that's because this vacuole doesn't have the amount of water in it should. So it's diminished slightly, so the cell is kind of not as plump as it typically is. But as you can see in your plant cell, you still have your nucleus, you have your nucleolus, you have your endoplasmic reticulum, both rough and smooth. You have your mitochondria, you have your Golgi complex or Golgi body. Now you also have, and this is again unique to plants, you have what's called chloroplast. And chloroplast is what helps the plant create its own food or use the process of photosynthesis to make its own food since they're autotrophs. All right, crew. Well, I think we're going to stop the video there for today. We've gone over what the history of the cell is, how scientists and biologists first started to see it, that use of that microscope. You've had a chance to see the difference between eukaryotic and prokaryotic cells. And now you've finished the front page, or front and a half of your notes, where you have drawn a completed and labeled animal cell and plant cell. Thank you very much for watching. I hope you find these videos useful to extend your learning and practice some more. Our next video is going to go into a little bit more detail on the organelles and their functions.